Michael, good morning, everyone. So today's our lesson is about fertilization. Okay, the whole process of fertilization. Okay, from the moment the sperm enter the vagina into the uterus, and what happens after that? Okay, all the way until the formation of zygote. Okay, so as the sperm, all right, enters the uterus from the vagina. Okay, capacitation occurs, right, which is the removal of the glycoprotein and plasma protein from the outer surface of the sperm cell, right, by the uterus enzyme. And this process is a part of the maturation of the sperm cell. Okay, so now after capacitation, okay, the sperm cell become mature, right, and then the sperm cell will swim all the way to the fallopian tube for fertilization. Okay, now in the fallopian tube, as all of you have known, okay, is the location where the sperm cell will um, come across or fertilize the secondary oocyte. Okay, so now fertilization is the fusion of sperm cell, all right, and egg nuclei, all right, producing a zygote that develops into new individual. Okay, so now you can see this diagram here, all right. So apparently this is the secondary oocytes, all right, and this is the sperm cell. Okay, so now in fertilization, okay, what happens first? Right, so first things first is contact. Now in contact, what happens? The sperm contacts with the follicle cell. So this is the follicle cells. All right, you see there are many layers of them, right? And these follicle cells, also known as the corona radiata, right? So the contact of the sperm cell with the follicle cells, right, will trigger exocytosis of the sperm's acrosome. This is the sperm's acrosome, right? So now, what happens after the acrosome is exocytosized from the sperm? Okay, acrosomal reaction will occur. Now, in this acrosomal reaction, what happens? All right, acrosomal enzymes, okay, are released from the acrosome. So you can see this acrosomal enzymes are released from the acrosome. Right? This acrosomal enzyme also known as hydrolytic enzymes. So, okay, the keyword here is hydrolytic. So hydrolytic means these enzymes will digest its uh, bahan to form substrate. Sorry. These hydrolytic enzymes mean to say these enzymes will digest its substrate. Okay. So the substrate, okay, is the zona pellucida. You can see the zona pellucida here, right? So these hydrolytic enzymes or acrosomal enzymes will digest the zona pellucida, and this zona pellucida also known as jelly coat of the secondary oocytes. Okay. Next, acrosomal process begins, okay, where growing actin filaments from the sperm heads. So meaning to say, actin filaments will grow from these sperm heads, right? Okay, will grow from the sperm head, okay, that will penetrate this zona pellucida, and then this actin filament, right, that protrudes from the sperm head, all right, it will bind to the sperm binding receptors that can be found in the secondary oocyte plasma membrane. So you can see in this diagram here, this is the secondary oocyte plasma membrane. Now, in or on the surface, okay, this secondary oocyte plasma membrane, there are many sperm binding receptors. Okay, so again, these sperm binding receptors will be bind with the actin filaments that generated from the sperm head. So I think I have the diagram here. Yes, so now you can see, okay, this is the actin filaments, right? that bind with the sperm binding receptor, okay, which is on the or in the secondary oocyte plasma membrane. This orange color here, this is the secondary oocyte plasma membrane, right? And this is the sperm binding receptor, right? This is the acrosomal enzyme that digested the portion of the zona pellucida, right? Okay, next, contact and fusion of sperm with the secondary oocyte plasma membrane. So the sperm cell must contact and fuse with the secondary oocyte plasma membrane. Again, this is the secondary oocyte plasma membrane. So now we are here, right? So what happens here is that the sperm will bind to and fuses with the secondary oocyte plasma membrane. So you can see in this diagram here, the sperm bind and fuse with the plasma membrane of the secondary oocytes. Right, so this fusion will triggers depolarization of the secondary oocyte plasma membrane, right? Some books says egg, so it's not egg, it's kurang tepat. So this should be secondary oocyte plasma membrane, right? So this fusion, the fusion of this sperm cell, the fusion of this sperm head, the fusion, let me rephrase that, the fusion of this sperm head with the plasma membrane of the secondary oocyte will trigger depolarization of this secondary oocyte plasma membrane, right? And depolarization of this secondary oocyte plasma membrane will prevent other sperm from entering the secondary oocytes. Okay, so if there is another sperm here that already digested the corona radiata, okay, the sperm cannot fuse with the secondary oocyte plasma membrane. If okay, there is another sperm which is earlier that has the sperm head already fused with the secondary oocyte plasma membrane. So other sperm that although they are in this part or any part, right, as long as there is an initial sperm or earlier sperm, okay, that has the sperm head already fused with the secondary oocyte plasma membrane, other sperm cannot fuse its sperm head with the secondary oocyte plasma membrane again. Okay, so the depolarization of the egg plasma membrane that prevent other sperm from entering the secondary oocyte, okay, this is also known as, okay, fast block to the polyspermy. Okay, it's an egg as a fast block to polyspermy. Polyspermy means multi-sperms. Okay, many sperms that can fuse with the secondary oocyte. So it wants to prevent this. And this is the fast block. So it will quickly, this depolarization of the egg plasma membrane by a sperm head, okay, will quickly prevent polyspermy, right? Quickly prevent polyspermy. So what happens after this? Okay, the sperm nucleus, okay, or some books says pronucleus, will enter the secondary oocyte cytoplasm. So currently, let me rephrase, after the sperm head already contact and fuse with the secondary oocyte plasma membrane, right? Depolarization occurs. Depolarization of this secondary oocyte plasma membrane occurs, which prevent entering of other sperm, okay? And then the sperm nucleus, right, of the sperm head that already fused with the secondary oocyte plasma membrane will enter the secondary oocyte plasma membrane. Okay, sorry, enter the secondary oocyte cytoplasm. Oh my, right? So you can see this is the sperm pronucleus. It enters into the secondary oocyte cytoplasm. So now we have the sperm pronucleus here, right? Next is cortical reaction. You can see this cortical granule here, right? Many cortical granule here, just under the secondary oocyte plasma membrane, right? So next is cortical reaction, okay, where cortical granules inside the secondary oocytes, right, or in the secondary oocyte cytoplasm will fuse with the secondary oocyte plasma membrane. You can see here this cortical granule fuse with the secondary oocyte plasma membrane, and this fusion will release, okay, enzymes. You can see these enzymes here, right? These enzymes will harden this zona pellucida, right? You see, zona pellucida. This whole thing here is a zona pellucida, and apparently these enzymes from the cortical granule will harden this zona pellucida. When this zona pellucida hardens, what happens? Okay, this will remove all the sperm binding receptors, okay, which is in the plasma membrane of the secondary oocyte. Remember, there are many sperm binding receptors here. And when this zona pellucida hardens, okay, it will remove all these sperm binding receptors. Now, after the sperm binding receptors are removed, right, fertilization envelope will form in the zona pellucida. Okay, so meaning to say the hardened part of the zona pellucida, okay, because of the cortical granule, is known as the fertilization envelope. Now, the formation of this fertilization envelope, okay, is known as slow block to the polyspermy. So initially, okay, the process quickly prevent other sperm from entering the secondary site, and the harden of this zona pellucida, or the formation of the fertilization envelope, will act as a slow block to polyspermy. Okay, so recall, this is the sperm binding receptors, which can be found in the plasma membrane of the secondary oocytes. Right, so all these are bye-bye, remove, dikeluarkan. Okay, now, next, what happens after fertilization envelope is already formed? Right, the secondary oocytes, okay, will complete its meiosis II, okay, to form an ovum, okay, and two polar bodies. So what happens after that? Right, sperm pronucleus, right, will fuse with the ovum pronucleus, okay, in a nuclear envelope, okay, and from here zygote is formed. Right, okay, so that's about it about fertilization, right, until the formation of the zygote. So in my previous lecture, okay, I already explained to you about oogenesis that occurs in the ovary here, right? And the outcome of the oogenesis is the secondary oocytes. Okay, and then just now I explain about fertilization. Okay, so from secondary oocyte, eventually, okay, it became an ovum with two polar bodies. This is second polar body. This is the first polar body. Okay, this is an ovum, right? And this is the ovum pronucleus, okay, that fuse with the sperm pronucleus in an envelope, and this will form the zygotes. So after zygote, we have implantation. Cleavage. Okay, from cleavage, and then embryogenesis. Sorry, after cleavage, implantation, and then embryogenesis. So next lecture we'll be covering about cleavage, morula, blastocyst, and implantation. Right? Okay, bye.